Organization and body plan. Levels of organization. Protoplasmic grade of organization. Unicellular organisms, all life functions are confined within the boundaries of a single cell. Protoplasm is differentiated into organelles. Examples: paramecium. Paramecium, where the protoplasmic grade of organization, where the single cell becomes an organism. It's unicellular organism. All life functions are confined within cell, where the organs or the different systems of your body are performed by different organelles of the protoplasm. So this is known as protoplasmic grade of organization. Paramecium, this lives independently, but it is a single cell, and the protoplasmic subunits, the protoplasmic units or the organelles, form the different organs of an organism. So such a complexity of life, that is known as protoplasmic grade of organism. The tissue level of organism, cell tissue grade. It is considered cell tissue grade, aggregate of similar cells that perform same function and forms tissue. You can't call it completely tissue grade, but here it is cell tissue grade. And some call the sponges as tissue grade. Sponges is the best example, which is the most primitive animal on earth. This exhibits cell tissue grade of organization. Tissue organ grade of organization, that is shown where different tissues aggregate to form organs, and these are shown by platyhelminthes. Platyhelminthes organs may have different types of tissues. They have eye spots, proboscis, reproductive organs that are organized to form a system. Organ system grade. Different organs work together to perform a function, forming a system. This is the highest grade of organization, and most animals belong to this grade of system level of organization. And the basic body plan. When you look all the metazoans, you are able to say that all metacellular animals, most of the metacellular animals, fall in 4 basic body plans: type of body, symmetry and degree of cephalization, the number of tissue layers or the germ layers, presence or absence of a fluid-filled body cavity, which is known as coelom, and the way the earliest events in development of the embryo takes place, that is fate of blastopore. These 4 characters are similar for all multicellular level of organization: the symmetry as well as cephalization, the germ layers, the coelom, as well as the development pattern. So based on complexity, it is further divided. Organisms are further divided, and based on symmetry and cephalization. Symmetry describes how the parts of animals are arranged around a point or an axis. Its presence or absence is a basic feature of the body plan. The animals which cannot be cut into 2 equal parts passing through any plane or axis are called asymmetrical. Their body lacks a definite form, and the body cannot be divided into mirror house by any plane. Most of the sponges fall in this category of asymmetrical one. 2 basic types of symmetries, radial and bilateral symmetry, are recognized in the metazoan animal body plan. Radial symmetry, arrangement of body parts such that any plane passing through the oral and aboral surface. Oral, where you have the upper surface, and the aboral, the lower surface, around a central axis will give identical halves, is known as radial symmetry. Animals with radial symmetry, they can interact with environment from any direction. Example are cnidarians, Ctenophora, that are primarily radial, as adults have been placed in the group Radiata. Bilateral symmetry, this is the symmetry that you find in most of the organisms, that where a single plane passes between the upper and lower body surface through the longitudinal axis of an animal, dividing the body in left and right mirror images, which is known as bilateral symmetry. This symmetry is known as bilateral symmetry, where a longitudinal axis cuts the body into two equal halves. Bilateral animals generally move in one direction, and the end that encounters the environment shows cephalization. The anterior end shows cephalization, which is the development of a definite head that occurs at the anterior end. This end encounters the environment first and develops the sensory, nervous and feeding organs. The other end becomes the posterior end. The back or the upper side is called the dorsal surface, and the front or belly side is the ventral side. Other regions are bilaterally symmetrical animals are lateral or the sides, medial or the midline of the body, distal end or parts that are farther away from the midline, and proximal parts which are near to the midline. A frontal plane actually divides the body into dorsal and ventral region, and the sagittal plane that divides the body into left and right sides. The frontal plane actually divides, and that is dividing the body into dorsal and ventral region, whereas the sagittal plane that divides the body into left and right sides. Animals that show bilateral symmetry, they are grouped as bilateria, and except for sponges, ctenophores and cnidarians, almost all animals including humans exhibit bilateral symmetry. Biradial symmetry, this symmetry is a combination of radial and bilateral symmetry. Here the body components are arranged with similar parts on either side of a central axis, and each of the four sides of the body is identical to the opposite side but different from the adjacent. This is seen in ctenophores, comb jellies with globular tentacles. Pentaradial symmetry, this is seen in echinoderms. Adult forms of echinoderms is another variation of radial symmetry, and most of these larval forms of these echinoderms exhibit bilateral symmetry, whereas the adult exhibit pentaradial symmetry, where the body can be divided into 5 equal halves through any plane passing through the oral and aboral surface. Spherical symmetry, this type of symmetry is seen in some unicellular forms and is rare in animals. It means that any plane passing through the organism's center is able to cut it into many identical halves. Spherical symmetry is best suited for rolling and floating and is seen in unicellular radiolarians and heliosovans. Germ layers and body cavity. Metazoans can be further divided into two groups based on the number of germ layers formed during embryogenesis. Germ layers, they are a group of cells that behave as units during early stages of development. These germ layers give rise to all the tissues and organs of the body of the adult. Animals that develop only two germ layers, they are known as diploblastic forms. They have an outer layer which is known as ectoderm and an inner layer called as endoderm. And the layer between these two layers, ectoderm and endoderm, may be filled with a matrix which is known as mesoglia. And two groups of animals, the cnidarians and the ctenophores, have this type of diploblastic development. Hydra, which is a cnidarian, we can see two distinct layers bound together by a non-cellular mesoglia, and the ctenophores also develop two germ layers. Animals from platyhelminthes to mammals all have triploblastic. They have a third layer which is known as mesoderm, which is present in between the ectoderm as well as the endoderm. The ectodermal tissue, that is the outer layer one, and the endodermal one, which is the inner tissue which forms a visceral organ. All animals possess some kind of body cavity, the most primitive type which is seen in poriferans and cnidarians. In sponges you have the spongocoel, which is always filled with water, whereas in cnidarians you have the gastrovascular cavity. Diploblastic animals have been further divided into three subgroups of body plans based on the presence or absence of an internal body cavity between the outer body wall and the gastric cavity. This cavity is formed during embryogenesis, and some triploblastic forms, thus body cavity is absent, and they are known as acoelomate forms. The space between the ectoderm and endoderm is solid and filled with mesodermal cells. The internal cavity does not form at all during embryogenesis. Some triploblastic animals have a fluid-filled internal cavity between the outer body musculature and the endoderm of the gut. This kind of space is termed pseudocoel, and the animals possessing it are called pseudocoelomate. Pseudo means false. The mesoderm in such animals does not surround the gut cavity but lines only one side of the blastocoel. Triploblastic animals that have an internal fluid-filled cavity lined completely by tissue derived from mesodermal cells are called eucoelomates. They have a true coelom which is derived from mesodermal cells. And this true coelom, it is formed of two methods. In protostomes, the mesoderm cells fill the blastocoel, forming a solid band around the gut, and by programmed cell that the space forms inside the mesodermal band, or there's a split that occurs within the mesoderm that results in a coelom, and this space which is formed is known as the coelom, and this coelom is known as schizocoelom, and the process is termed as schizocoely. Among deuterostomes on the other hand, the cell forms the center of the gut wall, begin to from the gut or the endoderm, outpushings occur into the embryonic blastocoel. These expand pouch walls forms a mesodermal ring enclosing a space. This space enlarges and merges to become the coelomic cavity, and this type of coelom formation is known as enterocoely, and the coelom is known as enterocoelom. This enterocoelom body cavity this is the first stage. Where the zygote divides into 2-cell stage. It further divides into 4-cell stage. 8-celled blastula is formed, and this blastula has a blastocoel. During early gastrulation, what happens is there's an internal movement of cell, and this forms a blastocoel. A blastopore is formed, and this blastopore, through this cells move inside the displace inside the blastula or gastrula, and there's a displacement of this blastocoel. So you get an ectoderm, outer ectoderm, inner endoderm, and somewhere you have the mesoderm. The newly found space within the endoderm is known as archendron. This is the blastocoel, and this blastocoel is getting displaced. This is the blastopore, which brings about the invagination of this gastrula. A fluid-filled internal body cavity between the outer body wall musculature and the gut that is lined by an epithelium known as peritoneum, which is derived from mesoderm. A peritoneum is there which lines the epithelium, and the organs of the body are suspended in the coelom by mesenteries formed by the peritoneum. A pseudocoelom lacks this peritoneum, whereas the true enterocoelomic ones, they have this peritoneal membrane. And the coelom provides a tube within a tube type of body plan for diploblastic animals, which provides greater flexibility when compared to animals that do not. The visceral organ is known as visceral peritoneum, and the peritoneal layer lining the body wall is known as parietal peritoneum. And development patterns. Diploblastic animals can be placed into two large groups, protostomia and deuterostomia. That's where stomia means mouth, and this is due to the blastopore, the presence of blastopore. In protostomia, the blastopore of the embryo forms the mouth and the anus develops on the opposite side. The pattern of cleavage in the embryo is spiral and determinate, where each is determined or destined to become special parts of the organism. Planes of cell division are diagonal to the vertical axis of the embryos, where the smaller cells are centered over the grooves between the larger cells below them. So that is the pattern. The developmental fate of each cell in the embryo is fixed, that is, each cell, even in 4-cell stage, if isolated, cannot develop into complete individual, but gives rise to a defective embryo if only one cell is lost. And platyhelminthes, nematodes, mollusks, annelida, arthropoda are the animals that are grouped and placed in protostomia. Among deuterostomia, the anus forms where you have the blastopore. There you have, in deuterostomia, you have the anus, and at the opposite end it is the mouth that forms later. It's radial and indeterminate type of cleavage that occurs indeterminate, where even if some portion is lost, the others compensate for this loss. You can note that the cells are aligned one on top of the other in radial one. Indeterminate cleavage means that each cell produced by the early embryo, if separated, can give rise to a complete well-formed embryo. The examples are hemichordates, echinoderms, as well as chordates come under deuterostomia. Segmentation. Segmentation is the division of body into smaller transverse compartments along the longitudinal axis of the body. It's a common feature of metazoan body plan. Each segment, it is called a metamer or a somite. True segmentation occurs in annelids, arthropods, and most chordates, though some other groups show superficial segmentation of the ectodermal body wall. Segmentation permits greater body mobility and complexity of structure and function. It divides the body into a series of compartments in which appendages, musculature, ganglia, nerves, blood vessels, coelom, and all other body organs are replicated. This arrangement is best seen in annelids, where it is considered as true segmentation. In chordates, segmentation is usually apparent in the axial skeleton muscles as well as in nerves.